Hello, everyone. Welcome to the session of Chapter 8 Cell the Unit of Life, NCERT Exemplar Part 2. So, today we are going to cover the Part 2 of NCERT Exemplar. Disclaimer. Learning objective. So, we will explore and understand the fundamental concept of the Chapter 8, the Cell the Unit of Life, by working through the NCERT Exemplar. So, while we are solving the NCERT Exemplar question, we will build up our fundamental concept related to this chapter, right? So, solving these questions are very, very important. So, NCERT Exemplar, that is Cell the Unit of Life. This is the index part, and here we will see there will be the multiple choice question, and the question number are from number 8 to number 14. That will be MCQs type. Very short answer type of question, that is question number 5 to question number 7. And short answer type question will be there, that will be question 5 to question number 8. And there will be some long answer type of question, that is from question 6 to question 10. So total 19 question we are going to discuss today. So let us start. Okay, so first question is here. Which of the following statement is true for the secretory cells? Secretory cells. This question is coming from the NCERT section 8.5.3, and the options are as follows. A. Golgi apparatus is absent. B. Rough endoplasmic reticulum is easily observed in the cell. C. Only smooth endoplasmic reticulum is present. And option D says secretory granules are formed in nucleus. Okay, so they are asking about the secretory cells. So let us see the correct answer of this question. So option B is correct. Yes. Rough endoplasmic reticulum is easily observed in the cell, and the cell that are secretory in function, that are related to secretory function. So rough endoplasmic reticulum is observed in the cells that are actively involved in the protein synthesis and secretion. Secretion means the secretion will be in the form of some proteins, like the enzymes are also, what is the chemical nature of enzyme? That is protein, right? So the protein is synthesized on a rough endoplasmic reticulum because the ribosomes are attached to the outer surface of the endoplasmic reticulum and makes the appearance rough. And that is why this type of endoplasmic reticulum is called as RER, or rough endoplasmic reticulum. The other options are incorrect as. So let us see why we are discarding the other three options. Golgi apparatus is present in secretory cells, right? They are also confined for the secretory functions. Secretory cell has both SER and RER. So both are there. There will be smooth endoplasmic reticulum, because the smooth endoplasmic reticulum, one of the major function is to make the hormones, right? Hormones are get synthesized in the SER, whereas the different enzymes related to the cell membrane, different proteins that has to be secreted out, they are formed in the RER. So SER is the major site for the synthesis of lipid, right? So lipid also gets synthesized in the SER. And no secretory granules are found in the nucleus. Nucleus is not related to any secretory functions, or any secretory granules are not found or confined to the nucleus part. So here you can see the endoplasmic reticulum, both you can see the smooth endoplasmic reticulum as well as the rough one. The rough one you can see they are having the this ribosomes attached to its surface. Okay. Okay, let us move to our next question. So next question is here. What is tonoplast? Tonoplast. And this question is coming from the NCERT section 8.5.3. Options are as follows. Outer membrane of mitochondria, inner membrane of chloroplast, membrane boundary of the vacuole of plant cells, and cell membrane of plant cell. So let us see what is the correct answer. So membrane bound to the vacuoles of plant cell. Vacuoles we see in the case of plant cell, and they are quite large, very large, right? Big size. And they got a membrane. This is a membrane bound organelle, vacuole. And they are given a particular name to the membrane that is called as tonoplast, right? So what is tonoplast here? Let us see that. In plant cell, the vacuole is bound by a single membrane called tonoplast, right? They are having single membrane. But in the cell, you can see there are many organelles that are double membrane. Double membrane. Can anyone give me the answer for this? Double membrane organelles. There are many organelles that are double membrane. Mitochondria, very good. Mitochondria is a double membrane, and also nucleus, chloroplast, very good. These are the organelles that are double membrane, right? Very good. Coming to the tonoplast. They transport ions and material against the concentration gradient, because what is the function of this vacuole? Try to understand that. Whatever the excretory substance, because you all know that the plant cell is not having, or even you can say the plant is not having any excretory organ. They do not have any excretory organ. So whatever the extra water is there, extra cell sap is there, extra material that gets synthesized, and they do not require them in the metabolism of the cell. So that all get stored it there, right? So transport ions, materials against the concentration gradient. Higher concentration of materials in the vacuoles than the cytoplasm. So materials that are having higher concentration, still they are present in the vacuole, but they do not come to the cytoplasm. So they are working against the concentration gradient. So organelle membrane, mitochondria, outer membrane made up of porine proteins, right? Very different, porine proteins. Chloroplast inner membranes surround the stroma, and these two organelles are having the double membrane. The cell membrane of the plant cell is not a tonoplast, right? That is a very typical type of membrane, and every cell is having, whether it is a plant cell or an animal cell, they all are having the cell membrane. So that is a very different type of membrane than the tonoplast. And you can also say that the tonoplast is having a very different structure, because they are working against the concentration gradient. They are basically meant for the storage of the waste substances that are produced, or the extra substances that are produced in the cell, right? So it is simple biological unit membrane present in every cellular organism. Which one? The cell membrane. Now here you can see the structure of the vacuole. This is the cell sap that is present inside the vacuole. There is a central vacuole, and you can see the tonoplast. The outer membrane is called as tonoplast. Next question is here. Which of the following is not true of a eukaryotic cell? Means which of the following statement is not correct regarding the eukaryotic cell? This question is coming from the NCERT section 8.5. And options for these questions are as follows. A. Cell wall is made up of peptidoglycans. B. It has ATS type of ribosome present in the cytoplasm. C. Mitochondria contain cellular DNA. And D. Membrane bound organelles are present. Peptidoglycan. So I got many of the answers. Let us see which one is correct. So let us move toward the correct answer. Yes, you all are correct. Very good. A option is correct. Cell wall is made up of peptidoglycan. This peptidoglycan structure we see in the case of prokaryotes. Yes, bacteria. Bacteria is a type of prokaryote, right? So prokaryotic cell wall is made up of peptidoglycan, not the eukaryotic cell wall. So what do you think about this? Ribosome in prokaryotic and eukaryotic organism are the site of protein synthesis, right? We all know that. Several ribosomes may attach to the single mRNA and form a chain called polysomes, and that translate mRNA into protein that is related to the ribosome. And the ribosomes in the case of prokaryotes and eukaryotes are different. 70S and 80S type, if you remember. The prokaryote is having the 70S type and the eukaryote is having the 80S type, and this is one of the options as well. So let us see the correct answer. The cell wall made up of peptidoglycan is found in bacteria and not in eukaryotes. They are not found in eukaryote. Instead, they are found in bacteria. And you can say in the broad term that they are found in the prokaryotes. Yes, prokaryotes. Eukaryotic cell wall is made up of cellulose, hemicellulose, pectin and chitin, right? Eukaryotic cell, it is made up of cellulose material, hemicellulose material, pectin substances is also there. Some of the organisms are having the chitin also. If you remember chitin, which organism cell wall is made up of? Chitin. Fungi. Yes, fungi. Fungi cell wall is made up of a polysaccharide that is called as chitin. Right. So again, the fungi is a eukaryote, not a prokaryote. So these are all the substances that here mentioned as a cellulose, hemicellulose, pectin, chitin, we see in the case of prokaryote, right? But the peptidoglycan, where we see the peptidoglycan? Peptidoglycan we see in the case of only and only in the prokaryote. So here the cell wall structure you can see, this is the structure of the eukaryotic cell wall. There is having the middle lamella, primary cell wall and plasma membrane, right? So this is the middle portion. Again, from the other side there will be the primary cell wall and then the plasma membrane of another cell, right? So this is the continuation. Thus is not true for the eukaryotic cell. Yes, peptidoglycan found in the prokaryote, not in eukaryote. In eukaryote, ribosomes are present in the cytoplasm, that is ATS type. So that was the correct statement. The ribosome in mitochondria, yes, in the mitochondria and chloroplast, both are having this 70S type of ribosomes. Yes, 70S type, because there is you can say a theory that is also called as Bose theory, though I'm giving this information apart from your syllabus. So it was assumed that the bacteria came into the eukaryotic cell, right? A bacteria came into the eukaryotic cell and it became the part of the eukaryotic cell. It get many modification as well, and that is now called as mitochondria. Similarly, the cyanobacteria, you all know that cyanobacteria or blue-green algae is again a type of prokaryote. It get enter inside a eukaryotic cell and it became a part of this. It got many modifications and now it is called as chloroplast, right? That is why this chloroplast as well as the mitochondria, they are having 70S type of ribosomes. Yes, there are many structures that we see in the case of mitochondria and chloroplast that are very, very similar to the bacteria and cyanobacteria respectively, right? Okay. So in eukaryote, the cell organelles are highly complex and the membrane bound, and the mitochondria in eukaryotes bear a separate circular DNA. Yes. We see in the case of bacteria that they are having the plasmid, small circular DNA, that is called as plasmid. That plasmid like thing, small circular DNA, is also present in the case of mitochondria and chloroplast, right? So again, this statement is true, but the first statement was incorrect. So let us take another question here. Which of the following statement is not true for plasma membrane? Not true for the plasma membrane. So this question is coming from the NCERT section 8.5.1 and options are as follows. It is present in both plant and animal cell. Lipid is present as a bilayer in it. Proteins are present integrated as well as loosely associated with the lipid bilayer, and carbohydrate is never found in it. So which statement is incorrect regarding the plasma membrane? So let us see the correct answer. So the correct answer is, yes, you all are correct. Option D, carbohydrate is never found in it. Plasma membrane, though the major components are the lipid as well as the protein, but yes, they do contain carbohydrate. Statement is false, right? This D statement is false. Why? The carbohydrate are present in the plasma membrane. They are present. They exist as a oligosaccharide attached to the protein, and that is called as glycoproteins. The proteins, suppose a small glucose molecule get attached to the protein, now it is called as glycoprotein. When glucose molecule get attached to the lipid, it is called as glycolipid. So glyco term is coming for the carbohydrate moiety that is attached to the either protein or the lipid part, right? And that is why it is called as, now this whole structure, glycoprotein and glycolipid, it is called as oligosaccharide. Okay. So do remember, the outer surface of the membrane, they do have glycoprotein and glycolipid, sometimes inner side also. So these carbohydrates face the outer surface of the membrane. Yes, I told you that majorly this glycoproteins and glycolipids, they are attached to the outer surface of the membrane. So they play an important role in the cell recognition and the cell-cell interaction, right? The cell and cell get interacted to each other. You all know that a mass of the cells or a similar type of cells unite together to form tissue. So there must be an integration. There must be cell-to-cell interactions, right? Some other vesicles that coming from outside get enter inside the cell. How they get enter inside the cell? Because cell-to-cell interaction, right? So here, the recognition of the cell, what type of cell is coming inside it, that is recognized with this particular glycoproteins or glycolipid. Now you can understand our immunity is dependent upon this. Yes, because any bacteria coming inside our body cells, how it get recognized, right? Though there must be cell-to-cell interaction, and which part is taking part in recognizing this glycoprotein or glycolipids, right? So this is very, very important, and this carbohydrate is a part of the plasma membrane, a part of lipid and proteins, right? So all other statement A, B, C are correct features of the plasma membrane. So here you can see in the diagram also you can see the sugar molecule. What is this sugar? Sugar means The carbohydrate, right? So here you can see the carbohydrate is attached to this lipid layer. The carbohydrate is again attached to this protein layer. This is the intrinsic protein. You can see the integral proteins or intrinsic protein that is traversing both the lipid bilayer. You can see here the sugar moiety is attached both the protein part as well as the lipid part, right? Okay, a very important part. Okay, now next question is here. Plastid differ from mitochondria on the basis of one of the following feature, right? Plastid differ from mitochondria on one of the following features. Mark the right answer. So you have to find out the right answer. This question is coming from the NCERT section 8.5.5 and the options for this questions are as follows. A. Presence of two layer of membrane. B. Presence of ribosome. C. Presence of thylakoids. And option D says presence of DNA. Let us see the correct answer. Option C. Yes, many of the students have given the correct answer. Very good. C option is correct. Presence of thylakoids. Yes, thylakoid structure is not found in the case of mitochondria, whereas the other structure we see both in the case of mitochondria as well as the chloroplast, right? Okay, so the structural element of the chloroplast differs than plastid from the mitochondria. What is that structural element? Thylakoids. They are the flattened sac stalked one above the other to form the grana. There are, you can see, like you can take the coins, you can take the 5 rupees coin or 10 rupees coin and you just put one above the other and make a pile of a coin. Similarly, the thylakoids in the chloroplast are placed one above the other. And there is a collection, there is a pileup of these thylakoids, and that particular structure is now called as grana, right? Now they are called as grana. So, what they are doing inside the chloroplast? The question is this. So, they are the site for the light reaction. They are the site for the light reaction, right? So, they are helping in the photosynthesis. If you remember this two types of reactions are taking place in whole, like photosynthesis mechanism. If you try to understand, it takes place in the two steps. The first step is the light reaction and the second one is the dark reaction, right? So this grana structure or the thylakoid structure is having what function? They are taking part in the light reaction. The stroma part, the liquid part that is present inside the chloroplast, and that is called as stroma matrix also. So that is taking part in the dark reaction, right? Rest of the features, what are the rest of the features? Presence of two layers of membrane. Yes, both the chloroplast and mitochondria are both two layers membrane. Ribosomes. Yes, both the organelles are having the ribosome. And what type of ribosome? 70S type, right? The presence of DNA are common to both plastids and mitochondria. Yes, both are having the DNA. Both are having the DNA in the chloroplast as well as the plastid. So this is again the correct information for both. But this thylakoid structure, you can see the thylakoid structure here given here. You can see this is one thylakoid, this is the second, this is the third, this is fourth, fifth, sixth. Now this is a collection of the thylakoids, right? So this is now this whole structure is called as grana, right? And there are some thylakoids that are attaching one grana to another. So that is called as stroma thylakoids, right? The thylakoid that is a part of the grana, that is called as grana thylakoids, and the thylakoids that are joining the two grana are called as stroma thylakoids. But this thylakoid structure is a very important feature of the chloroplast. It is not found in the case of mitochondria, right? So quickly, let us take another question. Which of the following is not a function of a cytoskeleton in a cell? Hmm? This question is coming from the NCERT section 8.5.7 and options are as follows. A. Intracellular transport. B. Maintenance of cell shape and structure. C. Support of the organelle. And D. Cell motility. Let us see the correct answer. Intracellular transport. Yes, this is a function of the cytoskeleton. So, an elaborate network of the filamentous proteinaceous structure present in the cytoplasm, that is referred to as a cytoskeleton. There are many tubules like structure, right? They are forming through a proteinaceous structure, right? And they are called as cytoskeleton. Cytoskeleton is not associated with the intracellular transport. The microtubules and the microfilaments are the component of the cytoskeleton and are responsible for the cellular and intracellular movement. Yes. So these are the two component, microtubules and microfilaments. So here you can see in the diagram, you can see the cell membrane and there are so many microtubules and microfilament. Generally, when you see the microscope inside the cell inside the microscope, you will not see such type of structure. You will not see. There are so minute and so delicate and so small structure that it is not visible with a normal microscope until and unless you see it in the electron microscope. There you can see this microtubules and microfilaments like structure. Okay. So rest of the options are the function of the cytoskeleton is a cell. Cytoskeleton provide mechanical support to the cell that help in maintaining the cell shape and structure. Yes, these skeleton-like structure means we are talking about the cytoskeleton. Like what they are doing, they are helping to make a particular shape of the cell and structure. The cytoskeleton also keeps other organelles such as mitochondria and ribosomes separated from one another. They do not, all the organelles do not get confined to one particular place. They are spread all together and they are like mitochondria and ribosomes, you will not see that they are present at the same place. Yes. So it also helps in supporting the cell organelles. But yes, they are also helping the organelles that are interconnected to each other, like endomembrane system. You have read about the endomembrane system. What happens there? The endoplasmic reticulum, Golgi body, lysosome, vacuoles, these are all structures that are interconnected. So here, these interconnection is done by this microtubules or microfilament like structure. Next question. The stain used to visualize mitochondria. If you want to see mitochondria, which stain you will use? This question is coming from the NCERT section 8.5.4 and the options for this questions are as follows. A. Fast green, B. Safranin, C. Acetocarmine and D. Janus green. Let us see the correct answer. So option D, Janus green is the correct answer. It is used to stain mitochondria, right? So if you want to see the mitochondria in a particular cell, then you have to use this particular stain. So Janus green act as an indicator and they change the color according to the amount of the oxygen present. Yes, you all know that oxygen reaches to where? Mitochondria, right? Because the cellular respiration is taking place at the site of mitochondria. So the oxygen, more and more oxygen is present there. So this stain is used as an indicator, right? And it change its color according to the oxygen present. And which organelle is having more oxygen? Mitochondria. So it oxidizes to the blue color in the presence of oxygen, and its absence changes to color to pink. Safranin, because this is again one of the option. It is used to stain nuclei and lignified walls of the cell. There are many organelles that are having the lignin substances deposited in it. So that for, if you want to visualize that, then you have to use this safranin solution. Acetocarmine, it is used to staining the nucleic acid and the chromosome. If you want to see the nucleic acid that is present inside the nucleus or the chromosome, then you have to use this particular strain that is called as acetocarmine. Fast green, it is used to staining histones. Histone is a protein, right, that is forming the nucleosome structure. So at alkaline pH, acid, after acid, extraction from DNA. Okay, now we will have a very short answer type of question. So let us take the first question. What is the function of polysomes? Coming from NCERT section 8.4.2. Let us see what is the function of polysome. So these questions are actually very, very important. It is not only that it is a structural type of, you can say that it is not a MCQs type of question, but yes, from this information, sometimes the MCQ question can be asked, right? So it is very important. Polysomes are the multiple ribosome attached to the single mRNA. There is a single mRNA, but many ribosomes are attached to it. So they appear a rosette or a helical arrangement. We will see the diagram just now, then you will understand what it is meant here. So each ribosome reads the same mRNA because the mRNA is same, na? So many ribosomes are attached it, and ribosomes are moving on the mRNA, so they are reading the same mRNA, right? So each ribosome read the same RNA to produce the identical polypeptides, right? So if you required a particular polypeptide in a large number, then there is a formation of polysomes. Function enable efficient simultaneous production of the multiple copies of the same protein. So here you can see here this yellow string structure that is representing the mRNA. This is the mRNA, yellow one, and you can see this gray color structure. This is the ribosome. So ribosome 1, 2, 3, 4, 5. So it is started here. So you can see here the small, this red color string like structure. This is the polypeptide. Again, you can see the polypeptide, it's longer than the first stage because it has read this much of the mRNA. You can see this third one, it is having the longer polypeptide because it has read from here to here, right? So in this way, multiple copies of the same polypeptide can be synthesized by the forming this particular structure, polysomes. Next question. What is the feature of metacentric chromosome? Metacentric chromosome. This question is coming from the NCERT section 8.5.10. Let us see the answer. So think about it before going to the answer. These are formed by the condensing and tight coiling of the chromatin fibers. Metacentric chromosome. When this particular type of chromosome, you see, this is a stage of a chromosome, right? So here the chromosome get highly condensed and tight coiling of the chromatin fiber. So the chromosomes are divided into different types on the basis of the arms and the location of the centromere. Very, very important. Where the centromere is present and what is the size of the arm. This is considered for naming a chromosome. And we are having the telocentric, we are having acrocentric, we are having submetacentric, and then we are having the metacentric. Now we will see the answer. The metacentric chromosome centromere is median. It is just in the center. Median. That is, the centromere lies in the middle part, middle portion, and thus forming the two equal arms of the chromosome. You can see the structure here of the metacentric chromosome. This is the centromere, and you can see this is the P arm, and you can see the Q arm. So P and Q arm are of the same size. Yes. So this is the metacentric chromosome. Next question. What is referred to as a satellite chromosome? Very, very important question. Satellite chromosome. This question is coming from the section 8.5.10. So the chromosomes may have additional construction or the secondary construction. So where is the primary construction then? The place where the centromere is there. That is the primary construction. But there may be another construction also in some of the chromosome. Right? In some of the chromosome, you can see a part of the primary construction. There is the secondary construction as well. So at their ends, as a distal part of the arm formed by the chromatin thread are known as satellite chromosome. So this appear as a small outgrowth fragment. And which of the chromosomes are having this satellite chromosome? Now here you can see this type of chromosome you can see as 13 chromosome, 14 chromosome, 15, 16, 21 and 22. These chromosomes are having secondary constructions. It means that these chromosomes are having satellite. They are called as satellite chromosomes, right? So again, the question can be asked, which group of the given chromosomes are having the satellite chromosome. So in the option 1, it can give 1, 2 and 14. In the option 2, it can give in 15, 16, 21, right? So in this way, they can give you the group of the chromosome. So if you remember which of the chromosomes are having the satellite chromosome, you can easily give the answer, right? So this is a very, very important information. So here you can see. This is one construction and this is the second construction. So these are called as satellite, right? Satellites. Next question. Differentiate between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So this question is coming from the NCERT section 8.5.3. So there are many differences. First of all, the structure-based differences. So thinking process before giving the answer. Endoplasmic reticulum is a three-dimensional, complicated and an interconnected system of the membrane like channels and run through the cytoplasm. Yes. And ER means endoplasmic reticulum was discovered by the Porter and Thomson. Additional information that you can remember. The Porter named is endoplasmic reticulum. So there are two types of endoplasmic reticulum, SER and RER. SER means smooth one, not having any ribosome attached to the outer surface, whereas the RER, the ribosomes are attached to the outer surface. Now, the actual answer, difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. First difference, RER possesses ribosome attached to its surface. Yes, SER does not bear ribosome attached to their surface. Second difference, mainly formed of the cisternae and the few tubules. So again, this is a structural based information. We are differentiating on the basis of the structure. So SER mainly formed of vesicles and tubules. Vesicles and tubules. And here cisternae and few tubules. Next, third difference. RER take part in the synthesis of protein and enzyme because the ribosomes are attached to it. So they will make protein, right? And SER, smooth endoplasmic reticulum, take part in the synthesis of glycogen, lipids, and steroids. Steroid means there are many uh, hormones that are steroid based. Yes, RER is internal and connected with the nuclear envelope. It is connected with the nuclear envelope, and SER is peripheral. It is present outside to the periphery, and it may be connected to the plasma lemma, or you can say the plasma membrane, right? So here we can see some other information. Yes, detoxification of the toxins in the liver and steroidal hormone production. Yes, smooth endoplasmic reticulum. We all know that glycogen, lipid, and steroid. So steroidal hormones are produced by the SER. Very good. So here you can see both the diagram. This we have already seen in one of the question. So some other differences. Fifth difference, it may develop from the nuclear envelope. So where it is developing RER, nuclear envelope. SER, it develops from RER. Sometimes the cisternae or the vesicles get cut off from the RER and the ribosomes are detached, and these tubules and cisternae, they moves towards the periphery and they form the SER, right? Sorry. Enzymes for the detoxification are absent in the case of RER. Yes, detoxification enzymes that are required, it is not found in the RER, but yes, enzymes that are required for the detoxification is found in the SER, smooth endoplasmic reticulum. RER, it forms lysosome through the agency of the Golgi apparatus. Yes, from the Golgi apparatus, these lysosomes are formed because the RER and Golgi apparatus, they are connected to each other. SER gave rise to the spherosomes. Very, very important information. So lysosomes are coming from the endomembrane system, where you can see the rough endoplasmic reticulum, the Golgi apparatus. See, the Golgi apparatus is mostly confined or near to the RER because the protein that got synthesized in the RER is transported to the Golgi apparatus to get modified, right? And it forms in a particular vesicle like structure, and it is pinched off from the Golgi apparatus. So sometimes the lysosomes are also pinched off from the Golgi apparatus and they are formed from this RER only, right? But the spherosomes, they are formed from the SER, a very important information, right? Next question. Give the biochemical composition of plasma membrane and how are the lipid molecules arranged in the membrane? So this question is coming from the NCERT section 8.5.1. Thinking process. To stay alive, all living things need membranes. Yes, every living organism is having a particular membrane. So membrane are the barrier which give cells their outer boundaries, that is the plasma membrane, and their inner compartment, organelles. You can see the organelles are also having the compartment, like they are having the outer membrane. So being selectively permeable, the plasma membrane control the movement of the substances in and out of the cell, right? The membranes are always selectively permeable, this plasma membrane, right? So they allow some of the substances to come inside and some of the substances to move outside, right? So it also regulate fluid composition, control flow of information, and finally involve in the capture and release of energy. Chemical composition of plasma membrane, let us see that. Lipid 20 to 79%, proteins that is 20 to 70%, carbohydrates that is 1 to 5%, and water 20%. It is highly variable. It is not for all the, it is not fixed. It varies from membrane to membrane. Yes, but most of it is having around 50 to 52% of proteins and lipid around 40%. So it is variable. So lipid are the major component of the cell membrane and they form the continuous structural frame of the cell membrane. So lipid can be of the phospholipid type, glycolipid, steroid. These are the different types of lipid. Yes. The lipid molecule possesses polar hydrophilic. So it is having the two types of poles. One is hydrophobic in nature, that is water repelling. So that is the tail part, right? And the one that is you can say the head like region, that is water loving. So the hydrophilic region that forms the head, while hydrophobic part that contain the tail of the fatty acid, it is inner side. So hydrophobic tail is present toward the center of the membrane and the structure results in the formation of a lipid. You can see here, this is the head and this is the tail. So the head is there and tail is downward, right? In this way they are present. So next question is, what are plasmid? Describe the role in bacteria. A plasmid is usually a circular, sometimes linear structure, double-stranded DNA that can replicate it autonomously. Why it is called autonomously replication? Because it can make its own copy. It can make its own copy, and this plasmid structure we see in the case of bacteria. It is a double-stranded structure. It is a double-stranded DNA. So these are found in the cytoplasm of the bacterial cell. Plasmid normally remains separated from the chromosome. Yes, this plasmid normally remains separated from the chromosomes. They are not part of the central chromosome, but sometimes may integrate into a temporarily and replicate with it incidentally. What is the role of plasmid in bacteria? Let us see that. Plasmids are the extra chromosomal. Why it is called extra chromosomal? Because the chromosome structure, chromosome DNA is different. Circular, long circular DNA that is present in the center, that is called as nucleoid structure, right? But apart of that, there are small circular double-stranded DNA that is called as extra chromosomal circular independently replicating unit, because it can replicate of its own. It can make its own copy beside the nucleoid. Nucleoid means the circular DNA. The circular DNA that is also called as chromosomal DNA. The plasmid is not the chromosomal DNA of the bacterial cell. Clear it is? So plasmid are used to transfer the information from one cell to another. So what is the use of this plasmid? What is the use of this plasmid? Plasmid is used to transfer the information from one cell to another. Means from one bacterial cell, the information is transferred to the another bacterial cell. So transfer the important genes, they may confer the resistance. We see that the bacteria is having the resistance against any antibiotic. So that resistance genes are present on this plasmid only. So this plasmid is very, very important. It also helps in conjugation of bacteria. Conjugation is a type of sexual reproduction that we see in the case of bacteria, right? So they form the sex pili. A pili-like structure means what is that? A tube-like structure that joins the two bacteria, and the chromosomes are transferred from one bacterial cell to the another, right? So this pili-like structure is formed by the genes that are present on the plasmid. So these days the plasmids are used in the variety of recombination experiment as a cloning vector. Now plasmid, we use this plasmid as a cloning vector in the recombinant DNA technology. You can see this is one of the plasmid structure, and it is a modified plasmid that is called as vector. You can see the ampicillin R means this is this pink color. Ampicillin resistance means ampicillin is a particular antibiotic, and the plasmid provide resistance to this ampicillin. You can see that tet R. Tet means tetracycline. Again, the tetracycline is a type of antibiotic, and if the plasmid is having this tet R gene, then tetracycline will not get affected. This antibiotic will not affect this bacteria, the one which is having this type of plasmid, right? Next question. What are histones and what are their functions? So let us see the answer. DNA is a polynucleotide, and one segment of DNA is called as gene. Yes, the DNA is a long polynucleotide structure, and many genes are present on this DNA. DNA has to be packed within tiny nucleus, and the DNA is very, very long structure, and the structure of cell we cannot see, so you can imagine how small the nucleus can be, and inside the nucleus a long DNA, the human DNA can be of 2 meters, and you can imagine now the cell that you cannot see, inside the cell the nucleus that is present you cannot see, how the 2-meter long thread can be present. Yes, so it is highly packed thing. The DNA is not freely in a form, it is highly packed. So how this packaging is done inside the DNA? How this packaging is done? Let us see the answer. Histones are the basic group of the globular proteins that have the high content of the basic amino acid arginine and lysine. So this histone is there, histone material, or you can say the protein is there. And what type of protein it is? Basic protein. These basic proteins are made up of the two types of amino acid, arginine and lysine. These arginine and lysine are the basic amino acid. So they are making the protein as a basic proteins, right? Histones are the basic group of globular proteins. Now what they do? Histone forms a part of the chromosomal material in the case of eukaryotic cell. This you can see, this nucleosome structure or the histone protein you see only in the eukaryotic cell. It is not found in the prokaryotic cell, right? So here you can see this round ball-like structure. This is called as histone octamer, and you can see the DNA. It is making a 2 circular coil on this ball, and here you can see one again, histone 1, H1 histone is there. So this whole structure is called as core of histone molecule, this green color ball. This is called as histone molecule, core of histone molecule. And it is also called as octamer. Why it is called octamer? Let us see that. So, there are 5 types of histone proteins: H1, H2A, H2B, H3 and H4. Now you take H2A, H2B, H3 and H4, each 2 molecules. So it will become 8 molecules. But the H1 is present as a single. So 4 of them, H2A, H2B, H3 and H4, occurs in pair to produce histone octamer. Octamer means 8 in number. So 8 histones, each H2A, H2B, H3 and H4 are present in pairs. So it becomes octamer. New body, it is also called as new body or the core of nucleosome structure. Histone bear positively charged end and they attract the negatively charged strands of DNA. Very important point. DNA is having negatively charged phosphate group. Remember, phosphate group is having negative charge. So DNA will impart negative charge on its surface, and histone is a basic protein, so it gives you the positive charge. So due to this negative and positive charge, they form this nucleosome like structure, right? So histone provide a medium around which the DNA winds, and they play a very vital role in the gene regulation as they act as a gene repressor. So they form the chromatin material and provide stability and are not coagulated by heat. And you can see this can get open. This coiling of the DNA structure on the histone octamer, that can get open, right? But this now do not get open because the H1 histone is attached to it. So it makes it closing it, right? So this is the structure of the histone, you can see here. And the 2-meter long DNA, it becomes so small that it can present inside the nucleus. It get highly coiled structure due to the presence of this nucleosome, right? So this is all about today's session, as we are having a very short of time, right? So this is all about today's session. Thank you very much for attending today's session. We will meet again. Till then, goodbye and take care.